Okay, so now we will move on to the next topic, that is the secondary growth in stem. Alright, so in stem of Cycas plant. Now in this diagram, it shows the secondary growth in the Cycas stem. Now secondary growth, it begins early in the young stem of Cycas, and at first it occurs in the same manner as in case of dicotyledonous stem. So the cambium which is present, okay, you see here, this is the in this vascular bundle, it is showing here at the side that this is known as the metaxylem and this is the phloem. So this cambium between the xylem and the phloem is known as the fascicular cambium. So the cambium present in each vascular bundle is known as the fascicular cambium or intrafascicular cambium. Now, as you can see here in this next diagram, here you can see that a new cambium strips are formed from the parenchymatous cells of the primary medullary rays which are present in between these vascular bundles. Remember, the medullary rays are present. So a new cambium strip will develop from these medullary rays in between these vascular bundles. So this newly formed cambium is known as the interfascicular cambium. So each newly formed cambium strip is called interfascicular cambium, and both the fascicular this fascicular cambium and the interfascicular cambium, as you can see clearly here, that they join with each other to form a complete ring of cambium. So they are joined together to form a complete ring of cambium, and this cambium, this complete ring of cambium, it cuts the secondary xylem towards the center. Okay, as you can see here, it cuts the secondary xylem towards the center and the secondary phloem towards the periphery. Alright, so besides this, cambium also produces a large number of the parenchymatous medullary rays. You can see here medullary ray in figure D. So this is the primary xylem and the primary phloem. Remember, the primary xylem and the primary phloem, but due to the formation of this new cambium strip, so they will cut the secondary xylem towards the center and the secondary phloem towards the periphery, and also they will produce these they will form these medullary rays. So these cells, they contain abundant starch grains, all right, these medullary rays. And now the activity of the primary vascular cambium, it will continue for few years, but later on it will become inactive and a new secondary cambium ring will develop. All right, so as you can see here, this is the first cambium strip, the first one here. So later on this will become inactive, a newly formed cambium strip will start to develop, all right, in the cortex or the pericycle. And this cambium, it will also produces the xylem toward the secondary xylem towards the center and the secondary phloem towards the periphery in the same manner as the first one. So you can see here also, this is the secondary xylem towards the pith and the secondary phloem towards the periphery. So both secondary xylem and secondary phloem, they are produced in the form of collateral bundles, and here the spaces in between these bundles, so these bundles, they are separated by broad parenchymatous medullary rays. And this cambium, all right, this second cambium also, it ceases its activity after few years and a new cambium ring will again develop. So you can see the first cambium ring, second cambium ring. So it develops in the same manner. In this way, successive cambium rings will develop one after another, and several rings of secondary collateral bundles, they are produced in the old stem of Cycas. So a 20 centimeter diameter stem of a species of this Cycas species, that is Cycas pectinata, it shows as many as 14 number, okay, 14 successive concentric rings of vascular cambiums. So in Cycas pectinata, and these concentric rings, they are separated from each other by a few layers of parenchymatous cells. But if you remember, we had studied in Pinus, annual rings are found in the are present in the stem. So here, annual rings like those found in Pinus stem, they are not formed in Cycas wood. Remember, in Pinus we have the annual rings, but here they are absent. And secondary xylem consists of the xylem tracheids, the xylem parenchyma, and also it consists the xylem fibers, whereas the vessels are absent. Right, the vessels in the secondary xylem are absent. That is the water conducting tissue of the plants. The types of cells found in xylem, the xylem vessels are absent, but some people, it has been reported that in some species of the Cycas, such as Cycas revoluta, these vessels are present in the secondary xylem. And the tracheids possess rounded bordered pits. Okay, the pits, they are usually found in 2 to 3 series. Sometimes they alternate or press. And whereas the secondary phloem, it consists of few fibers and also the sieve cells. Okay, so sieve cells, that is those elongated cells in the phloem. Just like in the case of Pinus, we had seen in Pinus, the phloem, they possess the sieve cells. And also some cells of the secondary phloem, it consists of crystals of calcium oxalate. Alright, so it consists the crystals of calcium oxalate, and some cells and the companion cells in the secondary phloem are absent. So here in the Cork Cambium, let us take a look at this diagram. Here the Cork Cambium, that is the phallogen, it develops from the outer layer of the cortex. Okay, so from the outer layer of the cortex, the Cork Cambium, that is phallogen, will develop. And this Cork Cambium, it cuts the cells of cork towards the outer side, and secondary cortex or the phalloderm towards the center. Okay, so outside the Cork Cambium, it will cut the cells of cork towards the outside and the secondary cortex towards the inner side, towards the center, and these layers form the outer periderm. Okay, it is known as, these layers together, they will form the periderm, and in the periderm the lenticels are present here and there in the usual manner. Okay, just like in other woods, the lenticels are present, that is the raised pores, those pores in the stem of the woody plant that allow the gas exchange between the atmosphere and the internal tissue. So just remember, in the periderm, that is the corky outer layer of a plant stem which form from the secondary thickening. So in this periderm, in this corky outer layer of the stem, lenticels are present. Okay, and these layers and the Psychostem is also characterized by the formation of the successive Cork Cambium, alright, which appear centripetally in the outer zone of the cortex. And also the outer periderm layer is peeled off, okay, as a result of the formation of next periderm below it. And this is the reason that the older basal part of the stem is comparatively thin, as the outer armor of leaf bases, they fall off. And looking at the overall internal structure of the Cycas stem, it becomes clear that the Cycas wood is soft and relatively spongy due to the presence of very wide parenchymatous rays, okay, the medullary rays. And the xylem portion is less as compared to the parenchymatous tissue, that is the cortex, the pith, and the medullary rays. And such type of secondary wood is referred to as monoxylic, okay, monoxylic. And such wood is commercially useless as and also generally associated with megaphilous leaves, okay, megaphilous leaves, and also the radial symmetry in the seeds of the plant. So that is all about the secondary growth in Cycas stem. Now let us talk a little bit on the characteristic feature, okay, just a summary to. Now the first point you have to remember is that, as you can see here, the outline section, okay, the outline of section is irregular. Why? Due to the presence of these leaf armor, leaf bases. And also the second point that we have to remember is the presence of large parenchymatous cortex as well as pith. And the cells, they also contain abundant starch grains, also known as sago. Then the vascular bundles, they are very narrow, as you can see, and they are radially elongated. And also the primary cambium is not well marked. And the third point is the presence of the mucilage canals, okay, in the cortex as well as in the pith, okay, the presence of these mucilage ducts or mucilage canals, these here, both not only in the cortex as you can see here, but also in the pith. Then the next point is the presence of girdle leaf base traces in the cortex. Remember here, these are the leaf trace, the direct and the girdle trace in the cortex. These are the leaf trace. Then also remember that the wood is monoxylic. Why? That is the presence of very wide parenchymatous medullary rays and also less xylem tracheids. So the xylem tracheids are very less. And also the annual rings, as in the case of pinus, cannot be seen here. Okay, they are absent. But the concentric rings of secondary vascular bundles are formed. Why? It is due to the formation of this cambium rings, and such condition is known as polyxylic. And the presence, remember, in the leaf traces here, in just before it enters into the rachis, here in the cortex, the vascular bundle is enlarged, but here present of pseudo mesage or mesage leaf traces, okay, which is a characteristic of ferns. And the last point that you have to remember, that is the characteristic feature of the Cycas stem, is the formation of successive phylogen, that is the Cork Cambium layers in the outer zone of the cortex. Okay, so these are the important characteristic features of Cycas stem.